I would like to talk to you today about neutrophils. Everybody knows that neutrophils are essential in medicine. They compose about 70% of the white blood cells in humans, and they're essential for any infectious problem. If people that have neutropenia or genetic defects in neutrophil function are very susceptible to infection. So this is a neutrophil that we isolated from a human healthy donor. Most of the work we do is with cells that we explant from patients because there is really no good model for neutrophil biology. And this is a neutrophil that you will recognize because it has this polymorphonuclear nucleus and these different granules that contain different proteins that are very good at killing microbes like enzymes and antimicrobials. And this is a movie where we took neutrophils and infected them in vitro with Shigella, a bacteria that causes dysentery. And you can see first that the neutrophil is very attracted to bacteria. Then it engulfs the bacteria. You will see now clearly this phagocytic event. And then all these different granules that contain, as I said, antimicrobials will fuse with the vacuole that contains the microbe and hopefully eventually kill it. There is also production of reactive oxygen species on the vacuole that probably also help in the activity. And this is a phenomenon that is called phagocytosis. It was discovered about 120 years ago. And it was already mentioned, my team showed a different way that neutrophils kill microbes and the formation of nets or neutrophil extracellular traps. So this is a stack of confocal images of neutrophils that we again activated in vitro. And we've stained with elastase, which is an enzyme that is specific to one of the granules. And if you look, for example, here on a neutrophil that has not been activated, the elastase is clearly in the granules. We also stained the cells with an antibody against one of the core histones or against a DNA dye. And you can see that in a non-activated cell that is exclusively in the nucleus, but all the components, some granular components and the chromatin that is the DNA and the histone are in these extracellular structures that we call nets. Now, we've done proteomic analysis of the nets, and they are surprisingly simple. Around 70% of the proteins in the nets are histones, and the rest is a very low number, around 30 different proteins that have antimicrobial activity, like cathelicidins or defensins. Now, the idea of a cell actually dying and releasing its own chromatin was novel in biology. The idea of apoptosis was that the DNA had to be cleaved in small pieces so it's digestible to other phagocytes. So one of the main questions that my lab has been addressing is how nets are actually made. So this is a movie of the process of net formation. This is, again, cells that were isolated from healthy donors. And these cells are incubated with two different dyes. Both of them stain DNA. One is permeable in purple. So all the cells here are neutrophils. You can see that the nucleus is, again, lobulated, typical for neutrophils, and there is a second dye that is in green that is impermeable, and it will only stay the DNA when the membrane, when cytoplasmic membrane breaks. And if I activate the movie, you can see that the nucleus first compacts and then eventually expands and forms these extracellular structures that are the nets. And I will tell you right now that these nets have been very important in several dermatological diseases that I will mention at the end. So this is a static image of the same process that I showed before. This is transmission electron microscopy. This is a neutrophil that has not been activated. You can see the lobulated nucleus and the different granules. Already 1 hour after activation, you can see that the nucleus is not lobulated anymore, but it's actually spherical, and there are more subtle changes in the intracellular membranes. 2 hours after activation, this is the chromatin, and this chromatin is in direct contact with the cytoplasm, and these structures that are here are the remnants of the nuclear membrane. We can tell that because we can do immunoelectron microscopy with antibodies, for example, against the nuclear pore. And 3 hours activation, this is a cell where the chromatin, or the relaxed chromatin, that already is decorated with different granular proteins, occupies most of the cell, and we believe this is a cell that very soon will explode and make the nets. So neutrophils, I haven't mentioned, have a very short life. They are thought to live less than a day in circulation. They have a life in vitro of around 6 to 7 hours. We make around 10 to the 9 neutrophils per day, which is staggering. So the experiments that we do in the lab cannot involve any genetics. There is no time to transfect them or transduce them. So all the experiments that we do in my lab is a little bit like trying to do biology of in the 21st century with the tools of the 20th century. So one of the things that is clear about this form of cell death of netosis is that it's very unique. This is cells that were isolated from the same healthy donor. We can activate apoptosis by classical means, and you can see that the neutrophils can go bona fide apoptosis. There is condensation of the chromatin and vacuolization of the cytoplasm. There is also DNA degradation, and here we are repeating experiments that were done many years ago. We also induce necrosis in neutrophils by incubating them with a pore-forming protein that we purify from Staphylococcus aureus. And here the nucleus is delobulated, but the nuclear membrane stays intact, and then there is never spilling of the chromatin to the outside. And this last form of cell death that is made in response to several different, both exogenous and exogenous stimuli, for example, microbes, but also autoantibodies or cholesterol. This forms this process that we now call netosis, which is a process to ends up producing nets. And one of the morphological characteristics is that the intracellular membranes are disrupted before the cytoplasmic membrane is compromised. So one of the first suspects that we had in the formation of net formation was the activation of reactive oxygen species. So just to remind you, neutrophils are extremely good. Every cell can do that, but the neutrophil is very efficient in converting oxygen to superoxide. And superoxide then dismutates to hydrogen peroxide, and then mieloperoxidase converts hydrogen peroxide into hypochloric acid. And both hypochloric acid, those of you that come from America will recognize it as Clorox. In France, it's called Eau de Javel. And hydrogen peroxide is an antimicrobial. Both of them you've used either on your skin or in your kitchen to kill microbes. They can kill microbes, but at very high concentrations. They're extremely reactive molecules. And we propose that the creation of this superoxide is important. This reactive oxygen species is important in net formation. And we had a series of experiments that I will not show you with inhibitors showing that if we delete the hydrogen peroxide, we inhibit net formation. And then we started our first medical collaboration to have genetic evidence that actually superoxide is important in net formation. So we isolated neutrophils with patients with chronic granulomatous disease. CGD are patients that have mutations in one of the subunits of the enzyme that makes NADPH oxidase. By now we've seen probably close to 100 CGD patients with mutations in different subunits of the NADPH oxidase. And the results are, as I shown here, this is traditional microscopy. The naive neutrophils look perfectly normal. You can see the lobulated nucleus and the granules. And when we activate them, these cells look very flat, because like in the movie I showed in the beginning, these cells are very efficient at phagocytosis. Phagocytosis is not compromised in CGD patients, but the result is that they cannot make nets. So this is the genetic evidence that NADPH oxidase is absolutely required for net formation. And in order to do a complementation experiment, we took the neutrophils from the CGD patients and incubated with hydrogen peroxide. If you remember, hydrogen peroxide is downstream of the NADPH oxidase, and because hydrogen peroxide is cell permeable, we can induce nets in this way. And in experiments that we did with the Children's Hospital in Zurich in a gene therapy assay, we also showed that complementing a wild-type form of the NADPH oxidase, we also complement the formation of nets. So this is the scheme, and I just said that NADPH oxidase is required, and then we suspected that mieloperoxidase was also required. Now, mieloperoxidase is an enzyme that is sometimes tested when people arrive to the hospital, and if the hospital has a machine that can measure mieloperoxidase, it will report it. And it turns out that 1 in 2,000 Europeans have an MPO deficiency. And we were stunned because it's clear that not 1 in 2,000 Europeans are immunodeficient. So we started investigating more closely, and it turns out that there are many MPO hypomorphs, that is, patients or people that produce around 60% of the activity of MPO. When we take blood from these patients, that make nets completely normal. And we went in a wild goose chase to identify true MPO patients, and it turns out they cannot make nets. This is one of the examples. We find one in Iowa, one in Belgium, and one in Berlin, luckily. And this is the nets of a healthy donor. By now, I hope you can recognize them. And when we look at MPO deficient patients, they really don't make nets. The nuclei remain lobulated. And interestingly, these patients that were MPO deficient were initially suspected to be CGD patients because of their symptoms. So in the process of netosis, I already mentioned, we conclude with the experiments and other experiments I didn't show that the production of superoxide and hydrochloric acid is essential in netosis. But that is in the first 20 minutes of the activation. So one of the things we also notice is that when we stain the cells and look very closely, elastase, which is a protease that lives in the granules, moves from the granules into the nucleus. And this is a non-activated cell, and you can see that the elastase, which is in red, and the myeloperoxidase, which is in green, are in granules. But after activation, you can see this purple rim around the nucleus, which shows that the elastase has migrated to the nucleus. So that makes us think that elastase was also important, and it would be, our hypothesis would be consistent that the migration of elastase would only happen in healthy donors, which is what happens. So if we tell healthy donors, we see the migration of the elastase to the nucleus, but in CGD patients and in MPO deficient patients, the elastase is kept inside the granules. And elastase is essential for net formation. This experiment is one of the few experiments I'll show you we did in mice. They are in elastase deficient patients, but they have a different phenotype. That is, they don't make neutrophils. So in wild-type mice, to demonstrate that nets are important, we took wild-type mice and elastase knockout mouse and infected with Klebsiella pneumoniae, which is a gram-negative bacteria that causes severe pneumonia. And this is a very high amplification of the alveolus of the lung. And I show you a scanning electron micrograph. These are the bacteria. There are many of them, and you can see they are completely covered in nets. And when we take an elastase knockout mouse, the bacteria are there, but there are no nets, although the recruitment of neutrophils to the lung of neutrophil elastase knockouts is completely normal. So elastase is also required for net formation, and one of the functions of elastase that we know is that they cleave histones into smaller pieces. This is an experiment. We take healthy donors and we see this processing of H4 and other of the core histones to a smaller size in 2 healthy donors. But when we take CGD patients and myeloperoxidase deficient patients, this processing doesn't take place. So the second effect after the activation of superoxide is the migration of protease to the nucleus, and in the nucleus there is histone degradation that we believe helps in the decondensation of the chromatin. Now, one of the effects that we saw is that there is a nucleus, that the nucleus loses its nuclear membrane. And a very talented postdoc in my lab, Borko Amulic, said the only other phenomenon in biology where the nuclear membrane disappears is during mitosis. So he proposed a hypothesis that some of the elements of mitosis would be important also in net formation. And he showed this by taking mouse that are deficient CDK6. CDK6 is one of the kinases that allows the progress of mitosis. And in naive, if we take neutrophils from these animals and we don't activate them, there are no nets. But if we activate either with a mitogen or with Candida albicans, we can see that the wild-type neutrophils make nets, but not the CDK6 knockouts. And interestingly, downstream of the CDK6 is tubulin. Tubulin is supposed to form this mitotic spindle, so chromosomes can migrate to the other sides, but neutrophils are terminally differentiated. And we can see that naive cells, these are non-activated cells, and the tubulin forms part of the cytoskeleton. But we activate the cell, the tubulin makes these centromeres, centrosomes that are required for mitosis. We don't know what is happening, why the centrosomes are required for net formation, because, as I said, neutrophils are terminally differentiated. So we require ROS, we require the migration of proteases to the nucleus to cleave the histones, and we also require the activation on certain parts of the cell cycle, and we believe this activation of the cell cycle is that what breaks the nuclear membranes and allows the expansion of the cell. So that still left us with this last event, which is how the neutrophils actually get out of the cell. And again, since we don't have a genetic assay, we decided to do a search for inhibitors. So in order to look for inhibitors of net formation, we devised a way that microscopically we could automatically measure the net formation. If you look at the pictures below, we can classify them in neutrophils that are lobulated, delobulated, diffuse net, or spread nets. And this is kind of a apologize for this, it shouldn't be here. This is kind of like a fax plot where we measure the perimeter of the nuclei versus the circularity. And in cells that are not activated, most of them look like number 1, that is lobulated, but 3 hours after activation, most of them look like nets. So this is the method we used, and initially we screened 2,000 donors, sorry, 2,000 inhibitors in order to validate the method, and then in collaboration with a company that is called Lead Discovery Center, that is part of Max Planck, we screened for almost 200,000 inhibitors. This sounds easy, but it was really heroic if you think that the cells come from a donor and you only have 4 hours to work with the cells. So these are the results. Around 10% of the compounds we tested were inhibited net formation by 50%, which is what we'd expect. And interestingly, we identify a compound that inhibited net formation when activated with a mitogen, with cholesterol, did not affect viability, and importantly, was not inhibitor of the NADPH oxidase, myeloperoxidase, and elastase, which we already knew were important in the formation of nets. And the inhibitor really works very well. This is the movie I showed earlier, where there is formation of nets after 2 or 3 hours. And when we incubate the cells in the form of inhibitors, the nuclei do compact a little bit, but they don't make nets even after 5 hours. So this inhibitor, turns out, it binds to gasdermin, which is a pore-forming protein that needs to be cleaved inside the cell in order to open the membrane. And we also showed that in net formation, elastase, the same protein that goes into the nucleus and processes the histone, also cleaves gasdermin and allows the formation of pores. So we still have many black boxes in the formation of nets. We believe that this research is important because, as I'll show you now, we think that nets are very important in different diseases, and it would be interesting to have possible interventions of net formation. So we think that nets evolved because nets are very ancient. Similar structure to nets exists, for example, in plants. They've been discovered afterwards, after we described it in humans. And we think their main job, they evolved because they are very good antimicrobials. One of their job is to bind to bacteria. These are neutrophils that we infected either with Shigella or Staphylococcus. And when we infect with either of the bacteria stick very strongly to the nets and prevent the dissemination of the infection. In fact, some of the gram-positives have evolved ways to inhibit net formation or secrete DNases to liberate themselves from net formation, and there are a series of experiments also with Staphylococcus aureus and with group B strep that are important in skin infections that show that this is an interesting effect. The second effect of the nets is that kill microbes. So this is a very simple experiment. We take neutrophils, we induce net formation, and then we wait for about 4 hours, so there are no neutrophils alive left, only nets, and then we add bacteria for 20 minutes. And we compare how many bacteria did we put in and how many bacteria survived. And when we do this in neutrophils where the nets are present, we can see here that in 20 minutes the nets kill in a culture around 25% of a Shigella culture or 35% of Staphylococcus culture. If we destroy the nets with DNases before we infect them, then there is no antimicrobial activity. And we've done this experiment in an alternative way, and that is to take neutrophils and incubate them with an inducer of net formation, and then with an antibody, an antihistone antibody, or with an irrelevant antibody. And when we then infect the nets that were already incubated with antibodies with a control, that were in a control antibody, we see again very robust killing of 3 different bacteria. The antihistone antibody prevents this killing. And we think this experiment has 2 possible interpretations. One is that the bacteria needs to be in direct contact with the nets to be killed, and the antibodies prevent this contact. The second is that actually histones have a very important antimicrobial activity. And this is a discovery that was done already in the late 50s. This is a paper by Jim Hirsch, then at the Rockefeller. And what he showed is that histones are extremely good antimicrobials. And the fact, if you measure the antimicrobial activity of histones, in the same molarity, histones are at least 100-fold more effective at killing both gram-positive and gram-negatives than for other proteins, for example, defensin and cathelicidins. So How histones kill microbes is something that also interests us very well. We have no idea. But one, we propose that histones actually evolved with two functions. One is to arrange eukaryotic chromatin, and the second one is to be an antimicrobial molecule. So the nets, as I already mentioned, evolved because they are important in immunity. They are very ancient, and they are important in preventing the dissemination and the growth of bacteria and viruses, parasites, fungi, and helminths. And for today's discussion, I think it's also important that staph and group B strep abscesses have been shown to be full of nets. Now, I already showed the process of net formation, but nets can be antimicrobial, but nets are also cleaved by DNases, which are present in circulation, and these pieces of nets can activate other immune cells like dendritic cells and macrophages to start a very strong inflammation. So the nets can also propagate inflammation. And also nets are important in coagulation. Nets are the source of the formation of a coagulose. This is the work that is mainly done by Denise Wagner at Harvard. So this immune activation is also important in the pathological side of nets. And nets have been now linked to many pathologies. So the production of nets at the wrong time and in the wrong place, or the lack of degradation of the nets effecting, have been linked to cystic fibrosis, thrombosis, lupus, and many other autoimmune diseases, arteriosclerosis. There is very interesting new literature showing that nets are actually promoting cancer metastasis. But there are two examples that I would like to just briefly mention, and was this hidradenitis suppurativa and psoriasis. And this is a paper that came from the lab of Mariana Kaplan at NIH very recently, and she proposes that in hidradenitis suppurativa there is a very important net component, and I just copied this image from her paper showing that in control skin there is not net formation, but in 3 different patients, in histological samples from 4 different patients, she stains them with both a DNA dye and citrunylated histone 3, which is a marker for net formation, and it's very clearly there are these structures present that are clearly important of nets. And in this paper, she has also information that destroying the nets ameliorates the disease. Psoriasis was one of the, actually it was Michel Gillier, if I remember correctly, who first proposed the role of nets in psoriasis, and there are now a number of papers in many different journals linking the neutrophil extracellular traps in promote autoinflammation in psoriasis, and there is an interesting new review on the role of nets in psoriasis. So with that, I'd like to finish, and this is an image, again, with a different DNA dye, an extracellular DNA dye, showing that neutrophils have the capacity under right circumstances to kill themselves in a specific form of cell death called netosis. This way allows the release of chromatin, decorated chromatin, into the tissue, and that nets have probably evolved because they have an antimicrobial effect, but if they are not dealt with in the right time and with the right means, they can also lead to severe pathology. And I already mentioned most of the people. Ilka Schulze and Renate Kruger are in the hospital surrounds my institute, are helping us a lot with immunodeficient patients. Herbert Waldman initiated the screen for net inhibitors and all the IL-cell coil-like. Volker Brinkmann is the head of our microscopy core facility, and he's been key on the net formation. And I should mention Veni Papagiannopoulos, who discovered the work on elastase, and Gabriel Solberger, who did all the work with the net inhibitor in our lab. And with that, I would also like to thank the Max Planck Society, who has been extremely generous and allowed me to do this work. And thank you very much for your attention.